So if you hear DNA replication, or if you hear the word replication, what is that? What is replication? And so I'm hoping you're saying something like making a copy, right? So replication is making a copy of something. And in this case, we're making a copy of DNA. Why do you do it? Why do you need to make more than one copy of DNA? You have to do it for cell division. There are two types of cell division, and each type has a different purpose. So the purpose of mitosis is to make new cells. So one reason you need to replicate your DNA is to pass that material on to the new cells. The other reason you need to do it is because you need to pass that material on to the cells that will become your gametes, okay? And that's the sex cells. So you need to do it to make identical copies of your cells, and you also need to do it so that your DNA can be passed on to your offspring. How do you do it? Oh man, okay. So don't get bogged down with all the details here. So one thing I want you to know, a lot of these words end in ace. So what does that say to you? It's an enzyme, right? Another thing I want you to remember is that DNA replication is semi-conservative. What does that mean? What does semi or hemi mean? It means half, right? So it's half conservative, half the same. So half of these come from the parent molecule, the original molecule, and then the other half is new. So if we look at this picture here, the old or parent is shown in blue, right? And then if you look at what you're making, so the new strands are half blue and half this orange color. So it's half old and half new. And that's what semi-conservative is. So each DNA molecule that you have is half of the original one it was made from, and then half of one that has been newly synthesized. Now, another thing I want you to keep in mind here is the basics of what's going on in this process. So the basics are this. First, you have to take apart this DNA molecule. So remember, your DNA molecule is this wound up double helix. The first thing you have to do is pull that apart. And you pull that apart by using this helicase. So helicase's job is to unzip genes. Okay, can we remember that? It's going to unzip genes. Now, another thing that has to happen is you have to make sure that that double helix doesn't form again. So you have these single-stranded binding proteins, SSBs. Their job is to keep that double helix apart. Now, on the front side of it, so ahead of the fork, you have this topoisomerase, and its job is to unwind any its job is to relax supercoiling. So if you think of DNA like a rubber band, and if you were to wind, like take your two fingers and rub the rubber band together, it introduced all these loops and whirls. So the same thing happens to DNA as you pull it apart. So you have to have topoisomerase go ahead and keep taking those things out to remove the pressure that will be placed on the double helix so that we can unwind it. Then you have to have this primase come in. So here we're looking at 4. This primase comes in. Its job is to lay the primer. The primer is a short RNA molecule, okay? Then you have this clamp that comes in. The clamp's job is to make sure that DNA polymerase stays attached to the DNA and doesn't keep falling off and having to come back on. So DNA polymerase is the enzyme that actually adds those new nucleotides, okay? And then you have ligase here. So ligase is at the end, and ligase seals our sugar phosphate backbone, okay? And now, so 2 other things you'll notice here is this leading and lagging strands. So our DNA has this orientation. So you see this 5 to 3, and then this one is this 3 to 5. And depending on which way your DNA is going to say which if it's the leading or lagging strand and how easy it will be to replicate it. So the leading strand is replicated continuously. The sliding clamp comes on, allows DNA polymerase to attach, and it stays on until it reaches the end of it. The lagging strand is not so easy. It falls off and comes back on, falls off and comes back on. And the reason being is that you see how distorted it is. You see how it's turned around. We have to make it look like it's in the right orientation. And in order to do that, we have to physically manipulate that DNA. And so it is synthesized in these short fragments of DNA called Okazaki fragments, okay? So short strands of RNA are the primer, short strands of DNA are Okazaki fragments here. So you have a leading and a lagging strand, you have semi-conservative replication, and you have a host of these enzymes, but pretty much 7 basic steps for DNA replication. This takes place during S phase of interphase, and that happens before mitosis and meiosis, and we'll talk about that in more detail as well, okay? Now, how does this work? So remember, this will be your parent strand. And when you pull your parent strand apart, you can then synthesize the daughter strand in the middle. But the same thing applies. A binds to T and C binds to G. So you can fill in what the replicated strands will look like. And a hint is it should look exactly like the parent strand you started from. Okay? So go ahead and take a few minutes and fill that in.